Great. Well, thank you so much for the introduction, and thank you again, Fernando and your team, for the kind invitation to share some of our work and our ongoing collaboration with the ALS community. So I'll start off by saying I'm not a clinician. I'm not a biologist by training. I'm an engineer, and I run a drug delivery lab at the University of Pennsylvania. And by drug delivery, I don't mean like a drug cartel. What I mean by drug delivery is we develop for drugs essentially what is a chemical GPS. So many drugs that we try to deliver in the body can't get to where they need to go, but by developing a delivery system, we can get them into places in the body that we need to in a safe and targeted manner. And the delivery system of choice that we utilize in the lab, as Fernando was mentioning earlier, is a lipid nanoparticle drug delivery system. So we develop next generation lipid nanoparticle drug delivery systems. Some of you might be familiar with these because many of you have taken them during the COVID-19 pandemic as these mRNA vaccines. As an engineer, I love these technologies because they're truly an interdisciplinary science feed. They're part RNA that encoded for the antigen of interest for the COVID vaccine. But what's unique is not only is it part RNA, but it's also part delivery system. It's part nanotechnology. They're made up of these nanoparticles that are about the 1,000th of a size of a human hair. And the delivery system in the case of the vaccine, this nanoparticle is essential for creating a vaccine. If you don't have the nanoparticle, the RNA cannot get into the immune cells by itself to create a vaccine. So we have these great technologies out on the market. These are a form of lipid nanoparticle with the mRNA. It's been developed by Pfizer, BioNTech, and Moderna. There are many people that came together to develop this interdisciplinary technology. Our friends and colleagues at Penn, Drew Weissman and Katie Carrico, they won the Nobel Prize for developing these mRNA technologies. There were many who developed the delivery system, in this case, the nanoparticle technology, such as Peter Cullis at the University of British Columbia. And then we had many people across the NIH and then BioNTech, Moderna, and Pfizer that scaled up these technologies at an unprecedented pace. And now that we have these technologies, a major question we get as a field is these have been great as vaccines, but by simply altering the sequence of the RNA, when we develop these, can we use these technologies to broadly revolutionize pharmaceuticals? Can we develop these technologies so they go into tumors to eradicate them? Can they be delivered into the heart to treat cardiovascular disease? And can they cross, in the case of ALS, can they cross the blood-brain barrier so we could then deliver therapeutic proteins to treat the disease? And there's certainly the potential to do so, but the key bottleneck, as I mentioned earlier, is the delivery challenge. So we have these RNAs. The RNA, the therapeutic, is a relatively mature technology, but the challenge is how do we get these delivery systems into the brain so we could express these therapeutics? And that's what my lab very broadly works on at the University of Pennsylvania. When I started my lab at Penn, we worked on this technology shown here in the middle. This is what we call an in situ CAR T cell therapy. So we're creating a cancer immunotherapy directly in the body. We design these nanoparticles in a way that they go into the bloodstream, they navigate towards the patient's immune cells, and then they re-engineer these immune cells into anti-cancer cells that eradicate the tumor. This technology is now in clinical trials for autoimmune diseases. But as a lab, we broadly think about this lipid nanoparticle technology as a platform. So by altering the construct, the lipid nanoparticle itself, its chemical composition, its formulation, we can now deliver to a range of cell type and tissue types in the body. I won't go over all of these today. I'm happy to talk about them later, but of course I'm going to specifically focus on our work with the brain. So can we develop something that goes beyond the blood-brain barrier to deliver into neurons? So how do we think about this as engineers in the lab? When we design these delivery systems, they need to cross numerous biological barriers to get to the cell type of interest. So if we think about this as intravenous administration, so if we administer something into the blood and we want to go past the blood-brain barrier and get to neurons, how do we do this? So we have an example shown here on the left. We first need to design the nanoparticles so they protect this messenger RNA cargo. So this messenger RNA cargo is like a very long strand of spaghetti. It's negatively charged, it's hydrophilic, it's a very fragile cargo. But if we condense it and we pack it into these nanoparticles that are less than 100 nanometers in size, we can now have them protected from different components in the bloodstream that would degrade the RNA. So we need to design it so it can navigate through the bloodstream. It then needs to cross these vessels. So in the case of the blood-brain barrier, this is a very formidable barrier because the pores are very small. It then needs to navigate through matrix and then ultimately get to, shown here in blue, the cell type of interest. Now, if we think of RNA by itself, if we wanted to get RNA into a neuron, it wouldn't get into the neuron by itself because, as I mentioned earlier, it's hydrophilic, it's negatively charged. The cells are negatively charged as well, so there's charge repulsion. But if we condense it into these small particles, as you can see here in blue, the RNA therapeutic of interest can then navigate into the cell through what are known as endosomal compartments. I won't go into this too much, it's a little bit complicated, but when we get things into cells and into these cellular compartments, we then have to release the therapeutic into the broader cellular cytoplasm in this case. So we design these materials in terms of when they get into these cellular compartments, they then become, instead of negatively charged, they become positively charged. And when they become positively charged and they interact with the cell membrane, it will then release the RNA cargo, and then the RNA cargo can be translated into therapeutic proteins to treat the disease. So to do this as a lab, I won't go into all of the chemistry behind it, but we develop what we call a drug delivery or a lipid nanoparticle toolbox to deliver these RNAs. My lab's a little bit different from a biology lab. We have what are known as material scientists. We also have bioengineers, and we have chemists that are developing these delivery technologies to deliver therapeutics to hard-to-reach sites in the body. Part of it is constructing the nanoparticle delivery system. So basically, very similar to Lego building blocks shown here in orange and in blue, we could build up different types of lipids that are the delivery system. And what we do is we don't just make one type of lipid, we usually make hundreds or thousands of these variants of lipids, because a lipid that works well for getting into a tumor cell or an immune cell is going to be very different than the lipid that works well for getting into a neuron or crossing the blood-brain barrier. So by making large libraries, we can now discover new lipids that can reach new tissues in the body. And this is ultimately a schematic of what these look like, these lipid nanoparticles shown here. So they're essentially these very small fatty droplets that are around 100 nanometers in size. As you can see here in the middle, they're made of 4 different lipid components that we utilize in the lab. There's an RNA component that encodes for the therapeutic of interest. And then we use a very tiny device that's about the size of a human hair, a microfluidic device, where we put all of these components in there and then mix them together, and out the other end comes these 100 nanometer particles. In the lab, we could do this in cell culture, we could do this in neurons, we could do this in a blood-brain barrier model, we could do this in mice. We also have the capabilities in the lab to scale this up to non-human primate studies, and then ultimately humans for clinical trials. So I'll talk about some of the work that we're doing specifically for delivery to the brain. We kind of have 2 angles for how we're looking at delivery into the brain. In collaboration with Children's Hospital of Philadelphia, we're developing local brain injections of lipid nanoparticles, and this is meant to be a one-time shot and cure for congenital brain diseases. So not so much for ALS, but if we have a congenital disorder that a child is born with, we basically take our lipid nanoparticle and we encapsulate CRISPR-Cas9 technology. And what we can do is inject directly in the brain, and we could edit out these deadly mutations after a child's born. We've done this in mice, we've done this in non-human primates, and we hope to initiate this further into clinical trials for various genetic disorders. But when we think about a local injection into the brain, it becomes very challenging needing to do that multiple times. And to treat ALS, you may need to develop a redosable therapy. So in collaboration with Fernando's team, we've been developing new types of LNP strategies for LNPs that could be delivered systemically and they can be redosable, with the hope that they cross the blood-brain barrier to deliver these RNA therapeutics. This is led by two very talented students in the lab, Emily Han, she's a PhD student, and Dong-Yoon Kim, who's a postdoctoral fellow in the lab. So how do we engineer these LNPs to ultimately cross this blood-brain barrier? So we have an example of what the blood-brain barrier looks like here. So as you can see, the endothelial cells that comprise the blood-brain barrier are very well connected. The pore sizes between these cells are very small, so it's very hard to get drugs across this barrier. It's also very hard when we develop new types of nanoparticles for drug delivery. How do we evaluate these for crossing the blood-brain barrier? So what Emily did was she developed a very cool blood-brain barrier model that we call this high-throughput screening blood-brain barrier, BBB. And basically, she's able to recapitulate the blood-brain barrier on a chip, so on a simple laboratory device. And what that allows us to do is we can now test very large libraries of formulations, hundreds or thousands of these formulations in a simple well plate. We could evaluate if the lipid nanoparticle crosses that model blood-brain barrier to transfect neurons. And then we could see if it works well in this engineered model, can we now recapitulate that result and validate it in animals to see if the nanoparticle crosses. So I won't go into all of the details behind this, but this is just a little bit of detail for how Emily developed this blood-brain barrier platform. Essentially what you do is you have one layer that is the blood-brain barrier, the endothelial cells, and then you have neurons underneath it. We could use microscopy to confirm on day 6. We form a very nice monolayer of endothelial cells, very similar to the blood-brain barrier within our bodies. And we could further confirm when we look at different types of molecules between the endothelial cells, the tight junctions, we could confirm their presence so we know we formed that tight barrier that prevents delivery of therapeutic cargo. We could then make these large libraries of these lipid nanoparticles and then determine the best ones that can cross the blood-brain barrier. So in this proof of concept study, we made nanoparticles. You could then label these nanoparticles with different fluorophores, and you could then transfect the blood-brain barrier model and ask, does it transfect the barrier? Does it cross the barrier, or does it transfect the neurons? And the results from this model were quite striking. So in this first proof of concept publication shown here, we were able to evaluate, I won't go into all of the details behind this, but in blue, in purple, and in green, you could see different types of lipid nanoparticles that we developed in the lab in blue and green. And then in purple, we compared this to nanoparticles that are developed by companies such as Moderna, Pfizer, and BioNTech. And what we were able to do, shown here in blue on the bottom, we were able to find various LNPs that cross the endothelial cells, so this model blood-brain barrier, and then also transfect the cells, which is very exciting. And this was really one of the first demonstrations of this. We could then take these nanoparticles from this model and then take the best ones and then ask, well, if we deliver them in vivo into mice, can we deliver these nanoparticles efficiently into the brain? So we basically injected these into mice. You could take out the brain and then confirm, have you transfected and delivered lipid nanoparticles to the brain? And we made this exciting proof of concept result shown here in the middle. If you look at LNP10 and LNP4, you see this greenish, reddish, bluish signal that indicates that the nanoparticle has gone into the brain, it has delivered its RNA cargo, and it's now expressing therapeutic proteins. So this was a very exciting proof of concept that we could develop a model to screen nanoparticles to see if they cross the barrier to get into the brain, and we could then validate this in animal models. And this is just further confirmation showing that we could evaluate these nanoparticles going into the brain. So they get into the brain, but then the question is, Where are they going in the brain? Are they going to the endothelial cells? Are they crossing into the neurons? As I mentioned, if we look at the endothelial cells shown here, they have these tight junctions that are present between the endothelial cells. And if you look at the nanoparticles shown here, you can envision it's very hard for the nanoparticle to squeeze through these pores. So what we've been developing as a lab is, can we develop nanoparticles that now have different types of targeting ligands on the surface, different proteins, and essentially through a lock and key mechanism, can we get the nanoparticle to bind to the endothelial cell and then transport directly through that cell to get to the other side into the brain. So we're developing a variety of these nanoparticles now that have these targeted peptides on the surface. I won't go into all of these, but there's a few of them shown here. One is known as RVG29. We have T7, we have angiopep, and then we have ApoE. We investigated a variety of peptides in the literature that have shown these potential properties that they could interact with the endothelial cells and then cross over that barrier. So we asked, can we leverage these targeting ligands to now ferry across the therapeutic RNA from the blood-brain barrier into the brain? And we have we did this first in this blood-brain barrier model, where we have the blood-brain barrier model and then we have neurons underneath it, and we asked the question, can these nanoparticles cross and reach neurons? And I won't go into all of the results here, but we're looking at endothelial cells, and then we're looking at neuronal cells on the right. The particle that we saw here in pink, this is with the peptide RVG29, we were able to show that this particle could cross the endothelial cells at very high levels and then transfect the neuronal cells that are on the other side. So the question is, how does this happen? How does the nanoparticle know how to get into the cell and then cross over onto the other side? So in addition to nanoparticles that we make in the lab, the endothelial cells in our body also make their own natural nanoparticles. These are called exosomes. So we ask the question, when the nanoparticle goes into the endothelial cell within the brain, does it get taken up by the endothelial cell? And when it gets transported back out, does it get transported in the form of these exosomes that come on the other side? They contain the RNA, and then do they transfect the neurons on the other side? So we did a very simple assay to evaluate this. We basically took a model blood-brain barrier, we put nanoparticles on that barrier, we washed anything that wasn't taken up by the blood-brain barrier, and then we monitored and then aliquoted anything that was secreted by that blood-brain barrier, and then seeded it onto neurons afterwards and asked, do these cells secrete the RNA and are then able to transfect neurons? And the result was quite exciting. This was shown here in the bar graph on the right. So if we see here in pink and blue, these two nanoparticles in particular get taken up by the endothelial cells, and then they get secreted as exosomes to transfect the neurons. And a variety of nanoparticles from this study shown here, and this is the case for all of the targeted peptide ones, RVG29, T7, angiopep, and ApoE, you see here in this blue, green, red signal that we're able to transfect the brain. We could even take out the brain as well and ask the question, well, it's not just going into the brain, but are we able to transfect the neurons in the brain? We could use a technique known as flow cytometry to do this. And basically what we're able to show here in red, this is in the second bar graph shown here, we're quantifying neurons. We see about 3% of the neurons in the brain have taken up the RNA and translated it into therapeutic protein. So these were very exciting results. We began working on this, and then we came into contact with Fernando because we were very excited about taking these technologies and utilizing them for different therapeutic applications. So I'll briefly mention this has been a really exciting project between Emily, Soyen, and Dongyun on my team, and then along with Fernando and Kyle Denton, and then the rest of the ALS TDI team. And the TDI team has been working on, very exciting therapeutic RNA cargoes at the time. So some of you are familiar with these therapeutic RNAs to potentially treat ALS. One is Stathmin-2. Stathmin-2 levels are reduced in ALS patients. And if we're able to restore those levels, so deliver RNA and express Stathmin-2, we could potentially utilize this as a therapeutic strategy for ALS. And then we also talked about this earlier today, C9orf72. So the C9orf72 mutation is a genetic alteration, one of the common causes of ALS. So we can ask the question, can we deliver the therapeutic functional form of C9orf using an RNA cargo? So we worked with them. Fernando and Kyle had these great therapeutic RNAs, and we asked the question, can we combine these with our LNP cargoes and see proof of concept? Can we transfect cell types of interest and then move forward with these assays? So we did a very similar type of assay as proof of concept here. We wanted to see in these model blood-brain barriers, can we essentially cross the endothelial cells and have the LNP deliver the therapeutic RNA encoding for Stathmin-2 or C9orf. And we've seen very exciting proof of concept results here. So we're looking at RNA expression of Stathmin-2 and C9orf after we deliver it to neurons using our lipid nanoparticle technology. So we're able to identify a variety of candidates. If we look here at the bottom, these are two LNP chemical constructs shown here. We identified two of them that were very promising to deliver to therapeutic RNAs from the ALS team. We were also able to show that one nanoparticle in particular, this compound shown here, C12306, not only transfects the neurons, but was able to show in the blood-brain barrier model that it could cross the endothelial cells and then transfect the neurons. So we were able to identify some of these exciting candidates shown here in these in vitro models, new LNP candidates that we could potentially utilize to transfect neurons. And we've been working with the ALS team on next generation LNP construction as well. So to develop something that's ultra specific, either for targeting the blood-brain barrier itself or for crossing the blood-brain barrier to getting into neurons, because potentially both could be of use. You could potentially leverage the barrier itself if you transfect it to secrete therapeutic proteins that could go in the brain, or you could utilize the neuronal specific LNPs to specifically transfect the neurons. And this is just a very proof of concept result where we're evaluating next generation constructs. But basically in both of these graphs in the middle, we're looking at mRNA delivery to either endothelial cells that comprise the blood-brain barrier or neuronal cells using 5 next-generation LNP technologies. And the result was quite exciting because out of the 5, we identified one that was almost exclusively delivering RNA to endothelial cells, so we could potentially utilize the endothelial cells as a protein secreting factory. But even more interesting, we found this candidate LMP4 shown here gets very little transfection of the endothelial cells. So it's simply crossing the endothelial cells. But once it gets into the neurons, we could get very high transfection of RNA. So we found that this is a very potent LNP for crossing the endothelial cells, transfecting the neurons. And we're now going to be leveraging this LNP therapeutic going forward to deliver a variety of therapeutic proteins that could be leveraged for ALS therapy. So in conclusion, as we think about this as drug delivery scientists, there are many opportunities for these LNP technologies, and we're really excited about their potential use for delivery to the brain. And by developing next generation technologies, we could develop something that's specific for the cell types of interest in the brain. We now have this model system where we could evaluate multiple LNPs simultaneously, very large libraries, to determine the best candidates and iterate upon that design. We've shown that incorporating targeting ligands similar to a locking key mechanism can enhance neuronal cell delivery. And we look forward to taking these technologies and continuing to work on them with the ALS team to deliver these therapeutic candidates, C9orf and Stathmin-2, and then push these forward into the in vivo models to treat ALS with these lipid nanoparticles combined with the therapeutic RNAs. So I mentioned many people throughout this presentation that did a lot of this work. So a big thanks to the ALS team for bringing us into this field, Fernando, Kyle, and others, to leverage our technologies to potentially be utilized to treat this disease. And then also Emily Han, Dong-Yoon Kim, and Soyeon, who have been doing great work in the lab with these lipid nanoparticle technologies and developing them for delivery to the brain. So thank you so much, and I'd be happy to take any questions.
Thank you, Michael. It sounds like we have a lot of great questions in the chat. Maddie, if you could go to Beth. She's going to ask a couple that are coming in online.
Great.
Okay, sure. Hi, I'm just going to read this directly, okay? Yeah. How is this research similar or not similar to the recent discovery by Sunnybrook researchers who made history in conducting a world-first study that has demonstrated the safety of using MRI-guided focused ultrasound to open the blood-brain barrier of the motor cortex in patients with ALS? They say that Sunnybrook is the only center pursuing this kind of work with ALS patients.
That's a great question. So I'm not familiar with this specific study, but I'm very familiar with the concept of ultrasound to improve delivery to the brain. So for those who may not be familiar, for therapeutic delivery, as I mentioned, across the blood-brain barrier, the pores between the endothelial cells are very tight, but there's been a lot of research broadly in the clinic for brain delivery that if we expose the patient to ultrasound, we could transiently open up the blood-brain barrier and increase the size of these pores for delivery. And this has been demonstrated for a range of applications. The idea is you only open the pores transiently. I'm familiar with the approach. There is a lot of evidence showing that this can improve transport of mRNA delivery, not mRNA delivery, just nanoparticle delivery in general. The one concern with ultrasound, though, is when you open the blood-brain barrier and then it closes back up, do you still have a fully intact blood-brain barrier, or has it been damaged in some way? And there is some evidence in the literature showing that after you open up the blood-brain barrier with ultrasound, it's not the same afterwards, and it could compromise the natural function of the blood-brain barrier. I don't know if this is the case for the mRNA study that's being done, but the motivation behind our approach is we're hoping to develop something that's hopefully safer. We don't have to modify the blood-brain barrier anyway or compromise its integrity, and we can let the delivery system do the work.
All right, we have a question over here to your right.
Sorry, there's a light.
You can't see people in the light. Yeah.
Yeah.
Great talk. Love the transcytosis system and the way to do it in vitro. Question on specificity. One of the things that would be great to have is directed towards a motor neuron instead of all the other neurons, because that would cause lots of non-specific reactions with it. Just curious, what's the work that you've done for targeting specific? Are there any targets that you've found on the cell surface that would help you target the motor neuron, or what's the status?
Yeah, that's a great question about specificity, and that's potentially one of the benefits of an LNP delivery system over, say, a viral delivery system that may transfect a large portion of cells in the body. So when we think about lipid nanoparticle specificity, we try to reach the brain. Of course, the brain is very challenging to reach, and the organ we're usually competing with is the liver. The liver is a natural sponge for nanoparticles. 90% of nanoparticles end up there, but we have tricks to reduce the response that we get in the liver. As we move forward with these technologies, what you can incorporate in the mRNA sequence itself is microRNA binding sites. And what happens is even if say you get a nanoparticle that goes to the brain and the liver and you're worried about what's happening in the liver, we could design the RNA so that it degrades when it goes in the liver, so we could reduce the off-target signal. So that's one way we broadly introduce specificity, but then how do we do that in the brain? There's two challenges that we really face in this area in terms of specificity. One is target identification. So that's something we don't do as a lab. So in terms of identifying unique biomarkers, For motor neurons, that's something we haven't done, but we would love to collaborate. If there's neurobiologists or clinicians looking at unique markers, our system is really a platform. We could introduce any peptide or antibody onto the LNP if we have that target of interest. But we haven't done it. It could be something that could be very interesting for transcriptomics or spatial transcriptomics down the line to identify these targets. The bigger question we have right now, and if anyone does this, we would love to collaborate in this area, is we don't really have good spatial resolution of where, once we cross the blood-brain barrier, what parts of the brain are we hitting? Are we hitting the neurons directly across the other side? Are we getting any sort of diffusion deeper in the brain? We don't have those imaging capabilities right now. We've talked to Fernando, and I think we'll collaborate and do some of this work in the future, but we would love to work with people on that. So the imaging, really getting spatial resolution, and then figuring out these targets would be very interesting.
All right, we have time for one last question.
Hi, thank you so much for your fascinating presentation that I mostly understood. So thank you. My question, is probably one of those ones you don't like to answer, but thinking of the ALS clock versus the ALS research clock, how long do you think before this technology will be available?
It's a great question, right? So normally I would say when we think of a non-viral nanoparticle technology, if we said this before COVID, the clock is usually very long to develop these technologies because you have to go through all the phases of the clinical trial. Patient enrollment is a challenge. But the FDA is now familiar with these technologies. These technologies, they're not the same exact lipid nanoparticle, but they're variants of those lipid nanoparticles. So they are very familiar with this technology going forward. I think the key next step for us, and this is something we would need to talk further with the whole community about, is we need to figure out how do we evaluate if we get the therapeutic and efficient concentrations, how do we evaluate that in the disease model? I think that's the next barrier we face. But from there, typically what we do after in the LNP field is we'll then do a safety study with a non-human primate to confirm that the LNPs aren't too inflammatory. And then after that, then it could then move forward into clinical trials. So it's hard to say right now. I'd say the key barrier, and this is something I don't have as much understanding because I'm not an expert in ALS, is how we evaluate this technology in the disease model and then go from there.
I think we're going to take one more question, actually.
Thanks. David Schechter, Albert Einstein College of Medicine. I'll make this a concise two-part question. So given the diversity of your LNPs, do you see A, difference in maintenance of expression of the delivered mRNAs, and two, any any differences in the immunogenicity of the LNPs?
Fantastic question. And the answer is yes. For both of those, we do see differences just by altering the delivery system. So mRNA, one challenge that we face is it induces transient expression. So very similar to the vaccines, if you deliver mRNA, if we transfect neurons, we'll see peak protein expression around 12 to 24 hours, and then we'll get an exponential decay. And then typically by the end of 5 days, all of the RNA will be gone. It will be degraded. So that's something to keep in mind for a therapeutic. You might have expression for a week, and then the question is, do you redose that technology afterwards, and how feasible is it to redose? So we're working with RNA, but one thing to keep in mind as well is this is a platform for a range of RNAs. You can take messenger RNA and circularize it and create what's known as circular RNA, and that could improve the expression for, say, 30 days, potentially. So we could then take, if an ALS patient can't get this therapy every week, but if they could go in for a monthly infusion, you could do this by modifying the RNA construct. And then the other question that you had is the immunogenicity. This is something that's a red-hot topic for the LNP field. The immunogenicity of the LNP in the vaccine is great, right? We want immune cell recruitment, but we don't want that in the bloodstream. We don't want systemic inflammation. So that's an active part of our LNP design. I'm happy to send it out. We published a recent paper. We haven't looked at it in the context of brain delivery yet, but now we can make LNPs that resemble the structure of olive oil, which we know has anti-inflammatory properties. And we're very excited to investigate these further for redosing at lower inflammation levels.
All right, thank you very much.